theme is nutritional epigenomics and age-related diseases. And first, I will give you an overview about the presentation. So I will talk about the DNA methylation and the one-carbon metabolism and how nutrients influence this methylation. And then we will talk about diet in aging and diseases. And I will explain how epigenetic clocks work. So they can measure the effects of environmental exposures linked to dynamic changes in the DNA methylation. And at the end, you will get a conclusion. So the first question is, what is epigenomics? Epigenomics is the study of the complete set of epigenetic modifications on the genetic material of a cell, known as the epigenome. The epigenome is involved in regulating gene expression, in development, tissue differentiation, and suppression of transposable elements. The genome itself remains logically static within an individual in comparison to the epigenome that can be dynamically altered by environmental conditions. So the DNA methylation is a big pattern of gene expression and the phenotype. It has different functions depending on its location within the genome. It is usually associated with transcriptional gene repression at CpG-rich promoters. So we've mentioned them yesterday, and this is a chemical combination of two nucleotides of cytosine and guanine. And the CpG sites dispersed throughout the genome are usually methylated. But the methylation also occurs at other regions, including transcription start sites and intergenic regions. The DNA methylation modifications are dynamic and extensively reprogrammed in early development and continue to a lesser but none of the less important role throughout the lifespan, owing to an influence of various environmental conditions, especially diet, which importantly contribute to the aging process and also to the disease susceptibility. So now I want to show you the one-carbon metabolism and the DNA methylation to just give you an overview because you can see that there are some nutrients which are important for the cycle. And like folate, B12, B6, B2, and also other nutrients like methionine, choline, and betaine, because they provide substrates and cofactors to help the efficient functioning of the system. And methionine, as you can see here in the middle, is regenerated from homocysteine and is a precursor for S-adenosyl methionine. And S-adenosyl methionine is then converted to S-adenosyl homocysteine. And a study showed that a supplementation with B2 and folic acid increased the mean plasma SAM. And the assumption is that the higher SAM-SAH ratio, the greater potential of methylation has the cell. And disorders in DNA methylation is often a result of a lack of bivitamins or also polymorphisms or interactions between the regulating enzymes. And a suboptimal B vitamin status is associated with increased aging of the brain, a declined cognitive function, and also with cardiovascular disease. So it is possible that there is a correlation between the B vitamins and the protecting role in age-related diseases. So the role of DNA methylation and diet in aging. The aging process is very complex and involves numerous changes at molecular and cellular level. Age-associated DNA methylation features in the middle age are identified in a large number of CpG sites, continuing to undergo changes into the old age. Especially environmental factors like diet, stress, physical activity, but also the socioeconomic status and smoking could impact the mechanisms or act directly to age methylome. And especially B vitamins may modulate the DNA methylation and thereby influence age-related diseases. And here I want to show you some study results because a study showed that DNA methylation of specific genes that are involved in the 1-carbon metabolism could induce the CVD risk, which is conferred by low dietary intake of B vitamins. And what other studies also showed is that supplementation with flavonols and polyphenols could also significantly impact methylation status. And for example, the epigallocatechin galate, which is found in a great amount in the green tea, is a key polyphenol that inhibits the DNA methyltransferase activity, resulting in a demethylation and reactivation of methylation silence genes in cancer cells. Other studies, which were quite interesting, with monozygotic twins, showed that although they are epigenetically indistinguishable between the early years of life, older monozygotic twins exhibited big differences in their epigenome. There is something like an entropic decay of DNA methylation during the aging, and repeat sequences generally become more hypomethylated during aging. Tissue-dependent DNA methylation variation may also explain why particular organs and tissues are more susceptible to different diseases. And many methylation changes leading also to inter-individual divergence during aging and are known as the epigenetic drift. And especially the CPG sites have been identified to undergo these procedures. And here I just want to give you an overview how this monozygotic twin study looks, because you can see that in early life, the twins have quite the same hypermethylation or hypomethylation. But when they get older, there are some differences. So in twin A, there is no hypomethylation in contrast to twin B. So this could also influence disease susceptibility and the aging of the epigenome. So now we come to the epigenetic clocks. Because chronological age as a predictor for risk of disease and mortality is suboptimal, because individuals with the same age may exhibit different diseases because of different aging processes, also as we've seen before. And this has led to several DNA methylation-based models of biological aging, which are known as epigenetic clocks. Each clock has a linear regression algorithm in the background that trains against the chronological age of the sample donors, and it selects a set of CPGs to produce a DNA methylation age that correlates accurately with the chronological age. Now just to say it again, what's bad about the chronological age: it's not a good predictor for risk of disease and mortality, and there are quite differences within the individuals. So now I want to show you the first clocks that were developed. It was the Harvard clock, which analyzed the methylation at 353 CPGs and was using a panel of 51 different non-cancerous tissues and cell lines. And this led to an accurate prediction of the DNA age across heterogene tissue and cell types. And the samples for the Harvard clock lead to a wide age range. So this is also an advantage, because also analysis of children and perinatal samples could have been made. The Hano methylation clock was derived from the analysis of whole blood in 480 individuals of either Caucasian or Hispanic ethnicity, using 71 CpGs to provide superior accuracy in the age determination. And a meta-analysis with 40,000 participants using the Horvath or the Hano clock showed that each 5-year increase of biological age leads to an 8 to 15% increase of the mortality risk. And when biological age exceeds the chronological age acceleration is said to be experienced, and this measure is of most interest. So, nowadays, there are recently developed clocks that have been trained on age-related disease and in combination with the chronological age. And two of the most robust one is the DNA phenotypic age predictor and the DNA-based biomarker of mortality GRIM age. The pheno age clock calculates the phenotypic age in a two-step process. So first, 42 biomarkers that predict mortality in the ENHANCE-3 study were used to derive an estimate of phenotypic age. And 9 of these biomarkers, plus the chronological age, were used independently of the DNA methylation to predict the phenotypic age. The Horvath and the Hano clock we've mentioned before are not influenced by the smoking status. However, the pheno age clock includes these disease-related factors. The pheno age clock was found to outperform the Horvath and the Hano age measurements with a more variety of aging outcomes because it also includes all-cause mortality, cancer, health span, physical functioning, and also Alzheimer's diseases. The most recent clock is the DNA GRIM age. It was trained using the Framingham Heart study, and it tracks the methylation of CpGs of blood-based protein biomarkers that are well known for health, like the PI1 or the GDF15. And to include the valuable informations led to improvements in the accuracy of age acceleration. And this measure is one of the most accurate ones in contrast to other traditional measurements like the telomere length, proteomic, transcriptomic, or metabolomic biomarkers. But what is also to mention is the clocks aren't linear during the lifespan. In studies, they showed impressive accuracy during the middle age. In later life, chronological age increased at a faster rate than the epigenetic age. And in childhood and teenage years, the patterns are also not linear because there is a greater rate of DNA methylation changes in children than in contrast to adults. So the epigenetic age and age acceleration are strongly linked to all-cause mortality, higher cancer risk, CVD mortality, and are also associated with important inflammatory biomarkers like C-reactive protein or IL-6. The epigenetic age and age acceleration is also linked to a variety of dietary factors like fish, fruit, and vegetable intakes, as we've mentioned in the Mediterranean diet. And they can positively influence epigenetic age acceleration. A study with omega-3 acid supplementation and vegetable consumption showed that they had a lower GRIM age accel. But what the study also showed is there might be a role of the sex because the omega-3 acid acceleration lowering was more pronounced in male people. And so this need to be careful consideration of the sex and the genotype and must be undertaken in the design of the epigenetic studies in the future. So the conclusion of the presentation is that despite their obvious strength, the DNA methylation-based clocks are unlikely to replace existing clinical biomarkers and measurements like blood pressure, the walking speed, or the grip strength, because they are very cost-effective and also easy to perform. But what is to mention is that the GRIM age is 61% more accurate than chronological age, and also 46% more accurate than the previously reported clocks, so the first well-known clocks, in predicting time to coronary heart disease. So, however, neither chronological nor GRIM age are entirely accurate estimators of coronary heart disease, and further work is required to determine their role as a predictor of cardiovascular diseases and other diseases outcomes. So thank you for your attention. And this is my source. So this is the paper if you are interested in, and I'm here for discussion. Thank you.
Ja, ganz herzlichen Dank. Das finde ich natürlich eine extrem spannende Geschichte mit dieser Biological Clock. Die Accuratness, also ich weiß nur, dass sie zum Beispiel in der Forensik, ich glaube, ich habe es gestern gesagt, durchaus auch schon eingesetzt wird in dem Sinn. Vielleicht um noch einen Link zu finden zu den Mikrobiota, kalorische Restriktionen als auch grobere Verschiebungen in Mikrobiota werden in diesen Clocks sehr deutlich angezeigt. Also hier gibt es offensichtlich einen sehr, sehr, sehr massiven Effekt, auch von Mikrobiota-Metaboliten auf die CpG-Methylierungen. Gibt es Fragen?
Ja, ich habe eine Frage. Du hast angesprochen, dass die Clocks ungenauer sind bei Kindern und bei älteren Personen und am genauesten bei Erwachsenen. Das heißt, ich kann die hauptsächlich dann eigentlich nur für Erwachsene verwenden, oder wie ist das zu verstehen?